name is Sabrina Templo. Today is June 17th, 2020, and this video is for Bio 225, Week 6 for the experiment Salt and pH Tolerance Testing for the results and reflection. When we talk about the salt toleration and the pH testing, we are talking about the limits that the microbes have in terms of their actual growth. So when we talk about microbial growth, there's a variety of different factors that you have to take into consideration, such as temperatures, which some temperatures like cold temperatures and some like warmer temperatures. Others, they like neutral temperatures in any wide range can cause bacteria to hinder from multiplying or to start multiplying. It really depends on what their limits are and what they do. Another is oxygen. So some microbes need oxygen to grow and others do not need oxygen. And if they have oxygen, they will die. Whatever the case may be, the same thing goes for pH. So if the pH changes to acidic, then they will die. And if it is too basic, then they will also have problems. For the salt tolerance testing, we are looking for the osmolarity, which is the interaction between the solutes and the solvents within the all-over solution. So an isotonic solution would be that there is an equal condition both inside the cell and external environment which surrounds the cell. A hypertonic solution would mean that there's swelling within the cell, which is based on the environment. A hypertonic solution would mean that there is cell shrinkage because of the exterior factors that is going around the cell. The other two things that we have to think about is the tolerance. If the bacteria is a halotolerant, then it means that it is okay to be in a salty environment. It doesn't necessarily need it to grow, but it can grow and it can live in that type of environment. If the bacteria is a halophil, then it needs salt in order for it to grow. So it cannot grow if it is not in a salty environment. So I have my vials, and I will start with my salt tolerance results. I have my S. epidermidis, my 1%, my 7%, and my 15%. For my S. epidermidis, as you can see, the 1% shows turbidity, which means that the S. epidermidis tolerates low concentrations of sodium chloride. And my 7% and my 15% are clear, which means that they came back with negative results, meaning that they do not tolerate higher concentrations of sodium chloride. And I did find that interesting because S. epidermidis grows on the skin and in very salty and warm environments. And I did find it interesting that the 7% and the 15% come back negative. My S. cerevisiae are also interesting because the 1%, as you can see, came back turbid, and the 7% and the 15% is clear, which is negative. So obviously, S. cerevisiae is unable to tolerate highly concentrates of salty environments, but 1% can. So it was interesting because this means that this yeast bacteria can grow in low salty environments. Next, I have my pH testing results. Just wanting to move that. Oops. I did find this pH testing interesting as well because typically the broth is at a pH of 7, and we use the HCI agent or the hydrochloric acid agent to decrease the pH and we use the NaOH, sodium hydroxide, to increase the pH. So I have my S. epidermidis vials and my S. cerevisiae vials. My S. epidermidis vials, for whatever reason, I'm not sure if it's my S. epidermidis culture that I have in my refrigerator is no longer potent, but my pH of 5 came back clear, or negative results for my S. epidermidis, which indicates that according to this result, S. epidermidis does not grow in a pH of 5, but my pH of 7 and 9 are turbid, meaning that S. epidermidis grows in a pH of 7 and 9. When we talk about pH, there are a variety of levels. There are acetophils that grow in an environment of a pH of 0 and 5.5. There are neutrophils, which like to be at a 7, which is half a point of the pH scale, which is 0 to 14. And they grow at a pH of 5.5 to 8. And the majority of bacteria grow are neutrophils. And finally, we have the alkaphils, which grow from 8.0 to 11.5 on the pH scale. So based on my experiment, the results, S. epidermidis are at the category of neutrophils and alkaphils. Next, I have my S. cerevisiae vials and 5 pH, 7 pH, and 9 pH, which you can see all of them are equally turbid, which also indicates that S. cerevisiae grows in a pH of 5, 7, and 9. So S. cerevisiae is a neutrophil that happens to have no limits of pH of 5 and very high limits of pH of 9. And I would say that they would just grow in a variety of environments since all 3 of my tests came back positive. I did think that this experiment was interesting, especially with the pH testing and using the HCI and the NaOH to manipulate the pH levels. And I did find it somewhat difficult that I had to keep using aseptic technique. But although I will say that I am getting better with it with each experiment. And that concludes my video for this week. Thank you.